So we are starting now. We are in the Regulatory Thursday. This is session 6. And we are going to talk about non-clinical and regulatory consideration to successfully get your molecule into the clinic, and some thought. We have learned from previous session that regulatory strategy impact on business strategy, indication and claims, classifications, pathway, regulatory or non-regulated, regulated or not regulated. So it's important for their product requirements, technical and market, the clinical trials, the competitive differentiation, the commercialization, who is the customer, who do you reach them. It's important for the reimbursement, the development timeline, the cost of development, the funding. So begin developing your regulatory strategy when you move from discovery to development or when licensing is paramount. So we have today two great speakers from Covance. Covance is by LabCorp is recipient of the 2020 Global Contractor Research Organization Company of the Year Awards. It's acclaimed by Professor Sullivan for its unmatched breadth of service to support clinical trials. And we have here Jessica Monrás, that is a successful inside sales manager at Covance with over 18 years of pharmaceutical early development experience, and have a lot of strong experience in fostering connection between nimble biotech companies and CRO subject matter expert. And we also have Dr. Baldrick, that is the Executive Director, Regulatory Strategy with Global Regulatory Affairs at Covance. He has over 25 years in non-clinical development regulatory support with wide cross-company experience in the development of many compounds, classes, and regulatory issue submissions. He has numerous peer-reviewed publications, over 50. He will explain more on that later. And is a regular podium speaker chairperson, as well as a lecturer on training course for a range of non-clinical regulatory topics. Professor Baldrick is also visiting chair, Regulatory Toxicology on the Lincoln School of Pharmacy, College of Science, University of Lincoln. He's a registered toxicologist and a fellow of the British Technology Society, as well as a member of various other scientific organizations. He's also on the editorial board of Journal of Toxicology, Food and Chemical Toxicology, Regulatory Toxicology and Pharmacology, associate editor of Board of Toxicology Report, is on the executive committee of the British Toxicology Society and is member of the Association of the British Pharmaceutical Industry, Preclinical Drug Safety Expert Network. So stay tuned and enjoy and acquire some regulatory expertise, because I think that is very worthy. So we will start with Jessica. Jessica, the floor is yours. Thank you so much for being here with us.
Gracias, Lourdes. Es un placer estar aquí hoy con vosotros. I'm so excited to be here today with you. So first of all, let's start by the toughest thing for me, which is sharing my screen. Okay. So let's see if you can see it. Can you see it, Lourdes? Is working well?
Perfect, in the right way. Yeah. So that's wonderful.
Okay, wonderful. Okay, so yeah, I'm very excited to be here with you today. When Lourdes asked me to, oh, sorry, now it's working well. Okay, so when Lourdes asked me to participate in this meetup, and she explained me that she's doing and the Transfer Knowledge Office, helping researchers with their projects and trying to build up those bridges between researchers and CROs, I thought, wow, it looks like if she has gone through my job description, you know, because this is exactly what I'm doing most of the day. So let me introduce myself and explain you a bit about my role at Covance. I am Jessica Monrás. I work as Inside Sales Manager for early development, covering Southern Europe, which includes Spain as well. I'm based in Barcelona. And although I joined Covance just 2 years ago, as Lourdes said, I've been working at early development for 19 years now. So part of my role as Inside Sales Manager is finding out and touching base with researchers who might have an interesting drug development project at an early phase. So what I do is I talk to them, I try to find out and understand what their needs are, so as I can involve the right experts at Covance and we can have further discussions and we can point those researchers on the right direction. Okay, so most of the times after following those, as a result of those discussions and conversations, we are able to put together a first quotation, a ballpark proposal, so as they can use it and they can include it in their business plan or finance plan, and they can share it with their stakeholders or investors, or they can even use it for a grant application. So having said that, I would like to share with you some slides to give you a very general overview of the standard hurdles you might find in the drug development journey and what the role of a CRO is. Okay. So I was told that you are a very heterogeneous audience today, but I understand your paths might lead you to create or at least to become part of a nimble biotech company someday. As a small biotech company, your role might be unique within the drug development industry. You could be a nimble venture moving quickly and economically with few layers of hierarchy, which is a real advantage, but you will also face unique challenges. For example, you might have limited resources. You will have to gain attention of stakeholders and managing the ever-increasing complexity of studies, and you will also have to keep your stakeholders confident and happy, which is not easy sometimes. Being a nimble firm, unfortunately, does not mean your drug development process is any easier than what the largest pharma organizations encounter. Actually, sometimes your questions are even more challenging and complex, since you're working on the next breakthrough in some of the toughest areas, for example, oncology, rare diseases, or biologics. Your program brings new questions, for example, companion diagnostics, changing regulations, fast-track designations, global approvals, or tackle recruitment of very rare patients. In the end, you want to avoid or unless successfully overcome any hurdles, because without the right plan, missed milestones, redundant activities, disparate data, and ultimately the loss of asset value can result. Your pursuit in drug development is challenging. Efficiently navigating the ever-changing landscape, managing risk and regulatory guidelines can be really overwhelming, and sometimes it's very difficult to know where to start. So always thinking with the end in mind, you have to advance from candidate selection to proof of concept and go through the rest of clinical phases to post-market, all while staying on track in meeting your stakeholders' goal. So I share this slide with you, but I don't want to spend time on this issue since my colleague Paul will talk about the drug development journey later. But I just wanted to let you know, for the ones who are not familiar with this, that the role of a CRO is exactly this: to come along with you in every single phase of the development and to help you to reach the milestones so as you can put your drug in the market successfully. Apart from CMC testing, bioanalysis, and regulatory support, which are services which will go along with you throughout the whole drug development journey, a CRO as Covance will also provide you with non-clinical services, which at the end of the day are those studies to be conducted before testing your product, your product in humans. At Covance, we have 12 of those non-clinical tox sites all over the world, as you can see in this map. And every project is so different that I'm not going to go deeper in the list of studies we conduct. Basically, as we say in Spanish, hablando en plata, these are any studies required by regulatory agencies before first into human. And all those studies, non-clinical studies required before later clinical phases, such as reproductive toxicology or carcinogenicity. So that's it. I would love to hear about your projects. So don't hesitate to contact me. Here you are my contact details. If you're not taking them now, you can ask Lourdes to put me through at any time. And I would be pleased to take a coffee with you, virtually, of course, everyone at her own place now. But I would like to talk to you, and so you can explain me where you aspire to go. And now, with any further delay, I'm pleased to hand over to my colleague, Professor Paul Baldrick, who will share some non-clinical and regulatory considerations to successfully get your molecule into the clinic. I hope you enjoy them.
Thank you very much, Jessica. Professor Baldrick, the floor is yours. Thank you so much for being with us.
Hello, everyone. I have to confess, I'm not a great fan of these virtual presentations. I like to involve audience participation. So you're going to get off lightly today, so I'm not going to be asking you any direct questions. But at the end of this, if you have any questions for me, I'm very, very happy to address. And I'm very happy to do this today. It's quite an interesting scenario. So I already have been introduced, so I'm not really going to go through this, other than saying that I've been in drug development for a long time now. And a little sell on Covance, my company. I've been with Covance about 20 years, and it's been a very exciting journey. And part of the reasons I enjoy working at Covance is every day I get to see new challenges. And you think you've seen molecules and development, but every day comes up with a new surprise. So it's a fascinating journey. Okay, what I'm going to cover today is some thoughts about your initial drug development considerations. And then we're going to go into more detail on the types of non-clinical testing you need to do. And then a few slides around how the difficult question of clinical starting dose, regulatory interaction, how you approach that. And then a couple of slides at the end about specific groups we have at Covance that can help you take your molecule into development. So getting it right. So a lot of this so again, the remit of this presentation, I understand that it's quite a variety of experiences of people on the call. So I'm trying to cover all of that. So within Covance, we work with a lot of startup, early and developed companies. And that can vary from one person to a number. Some companies are in the process of generating funding, while others have already secured backing up to a certain point of development. It might be to get the drug into the clinic, it might be to end of phase 1, or it might be beyond. So, and then others we deal with are established companies. So a bit of variety. And the other thing I certainly have learned in life is all companies have different approaches. One of the ones might be, you might be concentrating on actually generating a molecule to develop. So those would be companies, with a very much more academic slant, university background. So they're coming up, they're screening, they're coming up with a molecule to develop in a particular disease area. Other approaches, working on demonstrating efficacy. So you might be doing that in-house, a university, or with a small local company. And that's where you're working on showing your molecule actually has some sort of efficacy. And that might be in vitro models, cell-based models, or it might be some initial animal models, usually in the rodent. Then some companies want to have a bit of a bigger picture. They want the non-clinical studies, which I'll come on to shortly, types of studies they need to do to get the molecule into the clinic. And nowadays that tends to be done with a provider such as a CRO. And then we come across companies that want everything. They want all the non-clinical studies. They want us to help with the clinical. So that's where we would help them develop the clinical protocol for whatever types of, for example, phase 1 type study they might want. And we can support them with all the regulatory activities around that, the interaction with regulatory agencies. This is another important area. Again, have the awareness that companies we work with have very different experiences. And a lot of the companies we do work with are new to the drug development process. And I'm not being sort of disparaging, but sometimes we come across a scenario where the company or individuals we're working with are very, very fixed, or I've used the word stuck, in their own world. So they're developing their model, they're doing all these lovely studies, but they actually don't really know what they need to do with that molecule to get it further developed. And that's an area we you know, again, we've got good experience helping in that scenario. Another area is the company may have expertise in a particular area, but again, very specific. Something we've seen more so in recent years, a company may have a good experience with a small molecule, chemically synthesized molecule, but now they're moving on to biologicals. So biopharmaceuticals, which have got a completely different set of thinking and how to develop those molecules. So again, a lot of the experience you have around developing a small molecule isn't exactly applicable to a biologic. And finally, one of the areas we come across is you may not have worked with a CRO before. And one of the areas which is a key contact point between a CRO and a company is what we call the study monitor. So you're the person that is controlling the studies that are going out to the contract laboratory, but you may have no experience in how to do that. So again, we can coach and help you with that experience as well. Another one which often makes me smile is that some of the companies we work with are not really aware that we just don't do the non-clinical studies we do for fun. We actually follow regulatory guidelines, and although guidelines are guidelines, as I always say, you don't if there's something you don't want to do that's in the guidelines, if you've got a good scientific argument, that's often acceptable to the regulators. But there are sets of criteria out there that need to be at least generally followed. And those guidelines from a non-clinical perspective cover a range of pharmacology studies, ADME or kinetic studies, and toxicology. And again, as I say, I'll come back to more details on those shortly. Another shock to companies is that we need compound. We need sometimes a lot of compound, not as I've come across in the past, MIGs. We need grams of compound, and it needs to be properly manufactured. Okay, it can be small scale manufacturing, but the compound we test in our non-clinical studies needs to be as close to the proposed clinical form as possible. And this causes a great deal of discussion, delay, interaction with companies, because if you supply us with, as we call, a dirty batch of material, it could be full of impurities and have properties that are not necessarily representative of the material that we'll be using in the clinic. So if you use a dirty batch of material, you may end up with sort of adverse toxicology findings related to these high level of impurities. So we need to work with you to get material that's suitable. And two other areas which cause all sorts of interesting discussion. The types of studies we're talking about to get your molecule into the clinic cost money, unfortunately, and they take time. One of the areas over the years, I've come across this on many occasions, that there is can be a degree of nervousness working with, for example, a big CRO like Covance, in that we're not interested in working with you because you're a small organization. We're selling you studies you don't need. I can assure you neither of those should be concerns. In fact, I don't have the breakdown, and I'm sure Jessica can confirm this, but a lot of the companies we work with are in you call small area, individuals or very small companies. And to be honest, from my perspective, it's much more exciting working with a small company working on a new drug and getting involved with the science, as opposed to a big company that's just doing routine studies. So again, a bit of a misconception sometimes, and we certainly wouldn't be selling you studies you don't need, because it's our reputations that's at stake. Regulators get to see our work, and if we were performing studies that were not needed, we would certainly get come back from that. I put a couple of points up about using consultants. Some of the companies we work with use a consultant to help them. That's fine. We've got no issues with that. We often work with consultants. If you were thinking of using a consultant, my takeaway message too would be is make sure they have experience. We have seen many examples of individuals who have given advice where companies have gone off in the wrong track, doing studies that are not needed. And again, that results in wasted money, time. And to me, the biggest concern, and I've seen this again and again, you end up doing studies that are not needed with results which we find difficult to explain. And if you've got results that are difficult to explain, that makes your drug development pathway very, very, very well, uneasy to continue. And a typical example of that is using the wrong efficacy model and getting results that are not relevant to your molecule. And as I say, using the consultancy example, a lot of our activities within Covance are of a consultancy nature. We are actually working with you to get the best studies to the best design. So, the experiences we have are broad ranging and quite vast. Oops. Okay, so say you start working with us. One of the things we would do is initially is what we call a gap analysis. We'd look at the information you have and try and work around a pathway of what studies are needed. Unfortunately, sometimes the data we get presented is not great. I'm working with a company at the moment where it's an oncology molecule, and they've supplied me with about 20 or 30 Excel sheets of oncology model data and modeling, which unfortunately is not very useful. So again, we need to have data presented in a decent way. Further down the line, and this is something that's very important, and especially if you're in an academic institute, when you're getting your efficacy work performed, make sure you get study reports written up and signed, because later on, those studies will be needed for your regulatory submissions, especially for FDA. All studies that you perform have to go into the FDA's IND submission. And a number of occasions we've had to go back to companies, and they've had to go back to the institute where they've had their studies performed to try and get those studies written up retrospectively, which is not a good thing. So top tip on if you're working with academic institutes or you're in an academic institute, make sure you write up your studies when you finish them. Another area when we're working with companies is sometimes yes, sometimes no. They have an idea of what they want to do in the clinic. So they might have, and that is very helpful for us. So what the indication might be. Often when we're working with companies, if they're developing a drug for oncology, they may not actually know what they're going into, whether it's a life-threatening cancer or non-life-threatening. So again, both those sort of pathways have different thought processes into what you need to do. And I'll come back to that shortly. Whether you're going into healthy volunteers or patients, your dose route. I mean, most drugs tend to be oral, but obviously there's a variety of other dose routes as well, which might be of relevance. And if you've got an idea of how long you might be wanting to dose in the clinic, which helps us design the toxicology studies. Another area which we can help you, or which sometimes companies have an idea themselves, where do you want to start your clinical work? Do you want to do it in Europe? Do you want to do it in the States? Do you want to do it within one particular country? Do you want to do it, for example, in Spain? Do you want to do it wherever? So again, that's something we can work with you to help on that. And again, other companies are a bit more, I guess, development focused, and they have what we call a target product profile. So that's a high-level document covering what they want to do with the molecule, or indeed a clinical development plan where they cover all sorts of aspects on non-clinical development, their clinical work, early clinical work and beyond. And in fact, that's something we work with clients with. We can help you actually write your clinical development plan. I'm working on 2 or 3 of those at the moment. So it's something, again, that we work with companies to do. And another area of the development pathway all depends on the molecule you're developing. It might be a standard small molecule. It might be a small molecule which you're developing for oncology. It might be a biological drug. We work with a lot of monoclonal antibodies, or it could be something different. Obviously, the hot area at the moment is gene and cell therapy. We work with a lot of those compounds. We work with a lot of vaccines. We're working with a lot of companies with potential new COVID vaccines at the moment. So a lot of experience across all sorts of categories of drugs. And when you work with Covance, and Jessica alluded to this, it depends on the molecule, what you actually want from us. So obviously an initial contact would involve getting a proposal through our business development client services group, covering the type of work that you want doing. And the team then that you get, it could be a small team of non-clinical scientists if you've got 1 or 2 studies. It could be a larger non-clinical team if you're doing a whole array of studies, or indeed, a lot of the projects I get involved in, it can be a large team that covers both non-clinical, regulatory, and clinical. So variable approach as to how we handle things. Okay, so that's a bit of a background into the types of interactions we have with companies. And I'll say later on, I'm happy to answer any questions on those points. Okay, non-clinical testing. Going back to the outsourcing model, it can be everything. So every part of your drug development can be outsourced. And indeed, it's a paradigm we see with some companies. Others, it can involve the non-clinical, the non-GLP studies. So your pharmacology, ADME, and pharmacokinetics. It can be your GLP studies, and I'll come back to these shortly. It can be a whole array of support areas, your bioanalytical, your pathology, your regulatory activities, etc. And then again, it depends on the company's size and strategy. Nowadays, it's very, very rare for companies to do well, everything in-house. Back at 30 years ago when I started out in this industry, the companies I was working with, we tended to do everything in-house. But that's obviously changed now. So some or all of your activities are with a CRO. one of the paradigms we sometimes see is very early studies are done in-house, so the company can get a handle on their molecule and understand it better, and then bringing out the more involved studies to contract. And here's where you need to think about your checkbook. I didn't put together this information, but it's a study done a few years ago, and they reckoned that the average cost of all the activities, both CMC, pharmacology, metabolism, pharmacokinetics, toxicology, and your regulatory activities to get a molecule into the clinic are just over, on average, $6 million. So if that doesn't surprise you, you should get ready for that. And of those costs, in this analysis, about half of them were actually CMC. And then the toxicology, key toxicology costs were about 25% of the cost. So you can see that the types of activities we're talking about are quite cost intensive. Okay, just a bit of a breakdown now. Again, I don't know the background of those on the call, but I'll just, sorry for those that have a bit of experience, but it might be just a bit more of a training exercise. But for those that don't, the types of studies with a typical small molecule that you need to consider are obviously your primary pharmacology, your primary efficacy to establish if your molecule is potentially efficacious, and a biologically active dose. And that is becoming more and more important nowadays because we tend to use that biologically active dose in helping us set our clinical starting dose. And I'll come back to that shortly. Then you need to consider whether your molecule has got secondary pharmacology, i.e. off-target binding, and that can involve a whole array of in vitro binding assays, screening assays. Then we've got safety pharmacology, and that's where we're looking at a core battery of studies where we're looking at whether your molecule can have potential, again, effects on targets that are not necessarily what you're looking for, such as CNS, respiratory function, and the cardiovascular system. Then we've got our ABME, our kinetic studies. To get a molecule into the clinic, we need to do protein binding and in vitro metabolism studies. Also, on top of that, we need to consider our pharmacokinetic studies across species, usually rat and dog. And then nowadays, becoming much more important, we need to consider our drug-drug interaction. So what we consider doing is CYP inhibition and induction studies, and companies are looking at transporters as well, drug-drug transporters. Our general toxicology typically is 2 to 4 week studies in rodent and non-rodent, and that's where we measure assessment of exposure, which we call toxicokinetics. And we preface those studies with range finding work, make sure we get the right dose levels. Then we've got various genotox studies, typically an Ames test, or we call a reverse bacterial mutation assay, and another in vitro assay in mammalian cells. And sometimes we back that up with a bone marrow macronucleus study. And then on top of that, sometimes rarely, depending on the type of molecule, if it's got potential for phototoxicity, we might end up doing some form of in vitro assay to cover phototoxicity potential. And I don't want you to read this now, but it's a publication I did a couple of years ago where I was looking at the types of non-clinical studies we need to do to get your small molecule, your small molecule oncology, or your biological drug into the clinic. And you can see that there are different considerations and study types for these different criteria. So, for example, with a biological drug on the right, you wouldn't do safety pharmacology, or you wouldn't do genotoxicity studies. So again, it's just to give you a flavor of when you're designing your non-clinical package, we would take all these considerations into account. And just to give you a flavor, one of the things that we often get asked is, how many studies do we actually end up doing to get our molecule into clinic? And this is part of this survey I did a few years ago, where for a small molecule, if you look at the red highlight, typically we would run 35 studies, of which 16 of those were pharmacology, 12 were kinetics, and 9 were toxicology. So that gives you a flavor of that's why it's quite costly, in fact, doing a non-clinical program, because there's quite a lot of studies. However, if you're looking at a biologic, and then if you look at the red below, you can see that you're doing approximately the same amount of pharmacology studies to demonstrate efficacy. But because of the nature of these biologics, you tend not to do any or very little kinetic work and much limited toxicology work. So about approximately 50% less studies. Okay, sorry for this busy slide, but these are the sort of things that, so you start working with us and we're doing our first in human package. These are some of the things that we come across, which can give you headaches, delays, all sorts of unforeseen problems. Key one is getting us material to start the testing. And you know, we can put together these lovely Gantt charts with timings, which a lot of companies ask for. So this is when the studies are due to start, this is when they're due to end, etc., etc. But quite often we might come across a problem where you cannot supply us with the right amount of material on time. So that obviously all those studies need to be rescheduled. Then we run, say, a cardiovascular study, safety pharmacology, and we get QT prolongation in dogs or monkeys or whatever. What do we do next? Does this does this stop the whole program? Do we have to go back and evaluate it? So again, we have to take time to look at these findings. Or we do our genetic toxicology studies. Again, if we get findings in those, we may end up having to do further testing, which delays the program. We get severe toxicity findings. So say you get animals dying or all sorts of things. Again, that will need time delay and reevaluation. Similarly, if we get various target organ toxicities. I'm going through a program at the moment with a company where we've got findings in the testes of rats. Now, normally that's not too much of an issue if it's high dose, but for this drug, it's occurring at all dose levels. So it's not only seen in rats, not seen in dogs. So we're trying to work out, is it a rat-specific finding? And should we take that molecule into the clinic? Then with biological drugs, they've got their own specific problems. Quite often you get anti-drug antibodies because you're using a human protein in, say, for example, a monkey, and you can get immunogenicity findings. Sometimes death, you can get complement activation, which kills the monkeys, but it's all down to the nature of the drug, and you can still take that into the clinic. But again, we have to work through those findings. And I mentioned testes. Here's a couple we had recently. This is a drug was causing seizures in our toxicology studies at exposures which were close to what we predicted would be used in the clinic. What do you do? Again, this is where if I had audience participation, I'd be pointing to you in the audience and say, what do you think? So do we take it into the clinic? What do we do? Do we have to try and work out a mechanism? Is it a viable molecule? And then similarly, pancreatic toxicity, we had this recently with a molecule, again, very close to predicted levels we'd find in the clinic. Do we terminate that drug? And bear in mind, one of the issues here is we might be dealing with a company, this is their only drug. They don't have a backup. They're not like big pharma, which have hundreds of drugs in their pipeline. This is your only drug. You've invested in this a lot of money. So do you continue trying to develop it, or do you terminate it and start again? Very, very difficult decisions to make. Okay, a couple other areas I just want to highlight, again, which cause problems. If you get unexpected deaths, so say you're doing a toxicology study and you get animals dying with no apparent reason, or they get very marked clinical signs. So they're convulsing, they're sedated, whatever, but there's no obvious reason. We can't explain it because of the pharmacology of the drug. And the problem with that is you don't ignore it. We have to go through a very full investigation. Sometimes the conclusion is we don't know. We don't know why this animal died, but we need to make sure we investigate it properly. Now, the reason why that's important is because think of a human, even as a volunteer. So you take a healthy human subject and you give them the drug. You don't want them to die suddenly for no reason. So that's why you need to try and evaluate why this animal died in a toxicity study for no apparent reason. And again, there's ways around that. It does happen occasionally. So we'd maybe look at the bottom of the slide is lowering the clinical starting dose, looking at safety margins, and if it's an unmet clinical need, if it's, for example, life-threatening cancer, for example, pancreatic cancer, perhaps it's a less of a concern, and we can go into clinical trials with that concern. And one of the questions we get asked a lot, why do we do toxicology studies? Why are they important? And this is a nice survey that was recently performed from some Merck compounds, where over 100 of them, they discontinued development. And they discontinued development for approximately 60% of those drugs based on findings they got in toxicity studies. So again, that's why these studies are important. I mentioned clinical starting dose. This gives us great discussions. So we're working with you, we're working with the clinicians, we're working with the non-clinical people. Sometimes we're working with modeling people. We've got a group at Covance that does a lot of modeling from animal findings, your PK. So again, very involved area. One of the key measurements is what we call an OL. So again, if you're working on toxicology studies, what we tend to establish is an OL. And that's the level in the toxicology studies which we get findings which we don't consider adverse. So that might be the low, might be the mid, might be the high dose level. And these levels usually are, the low level is one that's close to one that we think we're going to use in the clinic. The high dose level is one, again, quite tricky to set. It's where we don't want to obviously cause animals to show great signs, but it's maximum tolerated dose. And then the mid-level is in between. So as I say, this NOAL from our toxicology studies is what we use to help setting our clinical starting dose. But on top of that, nowadays, we tend to look at a MABEL approach. And that's what we call a minimal anticipated biological effect level. And we also consider an estimated pharmacologically active dose. So we take a combination of our pharmacology studies and a combination of our toxicology studies. And occasionally, we might even look at our in vitro target findings as well to come up with a clinical starting dose. So my takeaway message from this slide is that's why you need to actually work with people who have experience in setting clinical starting dose, or you can end up with either an artificially low or an artificially high finding. And the paradigm, I don't want to spend too much time on this, I could go all day on it. Oncology drugs are a bit different because quite often you're going into people with advanced cancers and what they call limited therapeutic options, i.e. they're going to die quickly. So a lot of the oncology drugs are toxic. You get in a range of findings can affect, because obviously you're targeting rapidly dividing cells. So you can get a range of clinical findings across all organ systems. So what we tend to do there is we look at what we call a severely toxic dose in a rat, or a highest non-severely toxic dose in our dog or monkey, and we would work out a starting dose accordingly. Okay, just a few slides now to finish off. Regulatory interactions. So this is a group that I sit within. One slide on this, just to give you a flavor. Some companies are in a scenario, they've got a drug or a drug class that falls into a category of what we call fast development. And there are various initiatives within Europe and the US to allow you to get your drug quickly into the clinic. In Europe, it's called PRIME. So again, we can work with you and go through the process, the PRIME process, if it's applicable. So that would be allowing you to get drug into the clinic quicker. And similarly, the US have got various fast track, accelerated approval, priority review systems again, which again, we've got expertise within Covance to help you through that process if it's appropriate. So it's something to bear in mind, even early on in development, if it's a molecule you have that's appropriate. Again, regulatory process, not going through this in detail. The top blue line shows you the US process, where typically you have a pre-IND meeting, and then when you've got your drug through phase 2, you have a meeting, and then you have a meeting to get your marketing approval. And then the line underneath that tells you the types of documents you need. You need an IND, which is investigational new drug submission, and various other processes. And the end result is what we call an NDA, a new drug application. Europe's a little bit different. You don't tend to have as many fixed meetings, so you can have scientific advice whenever you want. And the first regulatory submission is what we call a CTA, a clinical trials application. And there's various other documents. Ultimately, you end up, after your 12 or 15 years of development, with an MAA, which is a marketing authorization application for regulatory approval. One of the questions we get asked is, should we have a regulatory meeting? I'm of the opinion only if you need to. So if you've got no strong burning scientific questions, so you've got no questions around the CMC, no questions on the CMC, on the non-clinical, no questions on the clinical, there is no point in having a regulatory meeting. All it does is add time, cost, delay. However, if you do think there are questions, and again, it's something we can help you look at and see if there are questions, we would then recommend a regulatory meeting. The nice thing about regulatory meetings, less so nowadays because they're not face-to-face because of COVID, is that regulators will say to you during the meeting, here are some other considerations for your drug development program. I mean, I've seen it on occasion. We go into a meeting, sit there, think it's a first-in-class molecule, and it's wonderful. And FDA say, sorry guys, we've seen one of these, or two of these, or three of these before. And they may and be honest and say, some of the considerations you've proposed may not be the ones that you want to do, and here's some further advice. But they will only say that in a face-to-face meeting, and it's not documented. So that's some of the benefits of actually having a regulatory meeting. Again, I'm not going to spend time on this because it's limited. But the types of meetings you can have in Europe are with what they call your local competent authority. So, for example, you go to your local authority in Spain. Here in the UK, where I'm based, we can go to the MHRA. Now we're no longer part of continental Europe. That makes it more difficult. So again, you can go to your local national authority, or you can have a meeting with EMA, which is called a scientific advice. In the US, various types of meetings. The second one down is the key one. That's your pre-IND meeting. That's also called a type B. Well, to say there's various other meetings you can have with FDA. Having been through this a lot, and I'm ongoing at the moment, setting up for a regulatory meeting, as I say, is time consuming. The process, as I say, you can ask for face-to-face, which you won't get at the moment. You can have a teleconference, which you can get, or written only responses. Basically what you need to do is send in a written request outlining your product and what questions you have, put together a briefing package with all the information you need, plan for a meeting, which you usually get 1 hour. Key is rehearsal and rehearsal and rehearsal. If you don't do that, you've got problems. And after the meeting, you'll get a written summary from the agency. Again, I'm not going to go through this in detail. It takes about, in time, maybe 2-3 months of development time for a regulatory meeting. And I've just put in some theoretical. So if you wanted to have a meeting early this year, you'd put a proposal in late March, then you'd get a response back from FDA or whatever agency saying, right, you can have a meeting, then you need to submit your briefing package late April, and then you'd have your meeting late May. So, and I cannot emphasize how important preparation of the briefing document is. That is quite a large, can be quite a large document. It's where you're summarizing everything you've got for your program, and you need to be clear because the questions you're asking, the data that you supply needs to be clear to the regulators so they can help answer those questions. So that's the regulatory process. Again, I'm not going to go into this. It's a group I sit in and I can talk for hours on this, but basically it's a cross-functional team. We've got expertise that gives strategy, where I sit. We've got a group that can write all the documents I mentioned. So you've got people who can write CTAs, INDs, briefing documents, etc. Clinical development plans, such like. And then we've got a group who can do all submissions. So as you know, if you're a small small company that have got no in-house regulatory capacity, we can make all the regulatory submissions for you, whether it's to Europe, US, etc. And that's covered by, we've got experienced regulatory management people. And then just to finish off, a couple of slides. Within Covance, we've got a group called Early Phase Development Solutions, EPDS. So I work closely with this group as well. So basically, some of the questions that Jessica posed earlier, these are the sort of things that we'd start off with, or within the EPDS group. What do we do with our molecule? Essentially, that's what all these comments on this box, on the slide say. What do we do? How do we get our molecule into the clinic in the best way? So this group essentially takes you through all the processes needed to get your molecule into the clinic, beyond the clinic, into proof of concept and beyond. And what do you get? So again, if this is sort of exercise or endeavor you would want with Covance, you'd get allocated what we call a drug development leader. So the little diagram on the left, that's the person in the middle, and they would interact with all the other activities, regulatory, non-clinical, clinical, and sometimes we work with companies that actually want market access or asset value, so we can put together a document to what your molecule has from an asset perspective. And they go through all the different aspects, supporting scientific advice, project management, etc. So I say that's a group we have in Covance. A lot of companies use it. And again, this is the sort of why it's important. The Gantt chart on the lower part is the sort of timings that this group can support versus the Gantt chart above, which is traditional timelines. So, and this is in-house data we've analyzed. It can save you up to about 30% of development time. So getting your molecule into the clinic quicker and beyond. So as I say, it's a well-used model we've got within Covance. And I think I realize that my time is more or less up. So that's the end of me. I'd be very happy to answer any questions. Thank you.
Thank you very much, Dr. Baldrick. I don't know if you have question, if you have any question or need further information, please feel free to ask here through the chat whenever is convenient for you. I know that there are some companies here that are developing some products. I have a question meanwhile. I say everyone is saying very interesting, very nice talk, but I'm looking for a question anyway. From the pediatric drug development, I mean, that is very specific. You have the possibility of juvenile animal testing, all of that. I don't know if it's needed or not. I mean, depend of the case, of course. But if you can give us some recommendation, because this is really even tougher than all of that. So.
Thank you. That's a great. In fact, it's one of my areas of expertise. I've published widely on pediatric development, non-clinical development. Um, it, it, it all depends on what stage you want to bring children into your clinical program. So most drugs you tend to develop them in adults. But within Europe, we've got a thing called a Pediatric Investigational Plan, a PIP. So that's where you have to go to the regulators, usually about phase 2A, where you have to go to the regulators and say, we're not going to develop this drug in children, or we are going to develop this in children. If they are going to develop this drug in children, here are the clinical studies which we propose to do in the near future. So you need to tell the regulators what those clinical studies look like and what age group you want to develop in. So it might be newborns, it might be from 2 years of age, might be from 6 years of age, it might be from 12 years of age. Those are the classic children age groups. So then the non-clinical side would come in then. Are there any signals from the available toxicology data you have to suggest that there may be extra data that a juvenile animal study would be needed to support those clinical studies in children. So, for example, there may be a signal that there's a CNS issue. There may be a signal on a particular target organ. In that case, we would have to do a juvenile animal study. We run a number of these at Covance, mainly in rats, and similarly in the US, there's a similar process. So it's called the PIP process, Pediatric Investigation Plan in Europe. In the States, it's called a PSP, a Pediatric Safety Plan process. So you go through exactly the same process with FDA, saying to them, this drug may be involved with children. Here's our clinical plans. And then you need to have agreement with them whether or not you need to do juvenile animal studies or not. So the whole pediatric things tends to come a bit more later in development. However, the slight difference, caveat to that, some companies nowadays are developing pediatric only drugs. So they are only being developed for children. So your first clinical studies tend to be in children. So that is a completely different paradigm. And again, we would have to work with you and think about what age we would dose the animals in the toxicology studies. So it may be from very young age, for example, dosing rats from 4 or 7 days of age. So.
Thank you very much. I'm happy because there are a lot of questions now. So we have first a tall one from Bob Jenkins from Inversine Research, that he's asking, what are the key factors to take into consideration to respond to a negative pre-IND response?
Okay. Hopefully, when you get responses back from FDA, they can be of two natures. One can be these are things you need to consider or want to consider, or other responses can be, these are things you need to address. So if I understand the question correctly, it's around how do we go back to answering these questions? Is that correct?
Sorry that I was speaking and I was in mute. So, Bob, if you want to ask something, I think that is what he means. But
I mean, the process of so basically if you submit an IND to the FDA, if they're happy with all the work you've done, all the non-clinical, if they're happy with the clinical, they come back within 30 days and say you can start your clinical trial. Often that's not the case. They come back with questions. So those questions can be non-clinical. So for example, they might want clarification on some of the findings you've seen in your toxicology studies. Can you clarify why you think these findings are not of concern? So you need to go back within those 30 days and respond to FDA why you don't think they're of concern.
Okay, that's perfect. Yes, he's saying that's correct. That's what he means. So perfect. He's
Yeah.
also Juan Carlos from Lino that is asking, they are working in a topical administration of our drug, of their drug, but we are not sure that is the best option. They know the drug also works in animal model through injection. Can we ask the agencies in a scientific meeting about this aspect, or is really a strategic decision of our company?
Fair enough question. My first question back is it for a topical indication, or is it being given topically to allow systemic exposure to develop a systemic disease?
Juan Carlos, can you speak?
Yes, can you listen to me?
Yes,
Yeah.
perfect.
Yeah. Hello. So the drug is now being administered through eye drops. So it's for a ocular disease, or it's actually a retinal disease.
Mhm.
So we know that it's working through eye drops in animal models. And we also know that it can work through intravitreal injection. So our doubts are related to the fact that translation of these animal studies to humans are not definitely straightforward. And we know that other companies have had problems with positive animal studies with eye drops. And when they have been performing experiments in humans, they were not successful. So our doubt is, should we go forward with our eye drops, or maybe lower the risk of the project and use our intravitreal injection instead? And I don't know if this is really a strategic question of the company, or if some advice can also be given by the agencies. I know it's a difficult question.
Oh, I'm not sure I want to be quoted on this answer. Yeah, I can see the issue with obviously eye drops is a much more favored clinical use than intravitreal. I mean, intravitreal is not pleasant. I think if you're confident that you're getting the same level of efficacy from eye drops, I think that's a process, or that's a pathway I would, pursue. Because obviously intravitreal, and also again, it's how frequently you need to apply the product, because obviously intravitreal's many weeks. Obviously you can't inject into the eye that quickly. Whereas obviously you can put eye drops in much more frequently. So I don't think it's a question the regulators would answer for you, to be honest. Going back to your original comment, I think you have to decide yourself and then provide the data to them showing that whatever pathway you take provides efficacious results.
And just as well, thanks for the answer. I know it's a difficult question, but as a follow-up question on this, another possibility we are trying to maybe to test also in the animals is a possible combination of both. Let's say you start your patient being treated with an intravitreal injection, and then you continue the treatment with eye drops so that you delay the following intravitreal injection. How easy is to do that? I mean, when you plan the drug development plan for preclinical and clinical studies.
Yeah, that's fair enough. It will make your non-clinical testing more complicated, because you will have to do, again, this is not knowing the molecule. I mean, if it's biologically active in rodent and non-rodent, you're probably going to have to do both species. obviously you'd probably have to do the in vitro injection, possibly in monkeys or rabbits or something like that. You might get away then with doing an eye drop study in the other species. So again, I'm just thinking off the cuff. You might be able to do an eye drop study in, say, rabbits and an intravitreal study in primates, and that could be your 2 toxicology species studies. So it could be as minimal as that, or it could be maximal. You know, you could have more studies. You might need to have 2 species intravitreal and at least 1 species topically on the eye. So yeah, going down that pathway, it's probably going to make your non-clinical testing a bit more extensive.
Yeah. Okay. Well, thanks a lot for the answer.
Thank you to both of you for the question and for the answer. So we have another question from Joan Montaner. Here they say, any advice for conducting trials on 2 repurposed drugs used in combination for a new indication? So 2 repurposed drugs used in combination for a new indication. I put also here.
Yep.
Joan,
okay.
if you have to add anything.
I can give you some good news and I can give you some bad news. There are guidelines out there. Both FDA and Europe have guidelines on combination drugs. And within those guidelines, they cover 3 scenarios. They cover 2 completely new drugs used in combination. They cover 1 completely new drug and a repurposed drug. And the third option is they cover 2 drugs that have already been on the market and are put together. And they give clear guidance on what you need to do. So in your scenario, if you're taking 2 drugs that are already on the market, you might end up with a small non-clinical package, or depending on how much information is available on the 2 marketed drugs, you could end up with quite a few studies. And unfortunately, that's something I can't go in. I can't give you any detail now on the call. That's something, again, we would have to work with you and see how much data is out there and what. But one of the studies that's commonly done is for example, if it's for a chronic indication, it's a 3-month rat toxicology study where you would have a dose level of each of the marketed drugs and then various dose levels of the combination. And the idea of that study is to show that you haven't got any new toxicity findings with the combination versus the individual drugs by themselves. So you could be talking about a 3-month toxicology study, but those considerations are all given in those 2 guidelines I mentioned on combination drugs. But it's certainly something we can help you with. And again, it's the sort of studies we would run at Covance. Does that help?
Yes, he's saying that thinks that is very useful, so he will probably follow up with you or later on or whatever. So we have also another question. Here he's from Antonio Sanchez Pozo. He's dean from the Faculty of Pharmacy and Biochemistry, and he teach a lot to the master students. And he's asking that if you can give us any particular advice for master students interested in the field. If they're master students, what do you think that will be the thing that they have to start learning? Or how should?
Well, one of my hats is I'm a visiting professor at a university in the UK, and I actually lecture on their pharmacy degree course. So I lecture to 1st, 2nd, 3rd degree. I also help out the PhD and MSc students which are doing a pharmacy background. So my advice to you is get as much toxicology training into your curriculum as possible. One thing we're finding, you know, it's a great advantage for
yes,
future work. If you're going,
toxicology. There is toxicology.
it is a toxicology.
Yeah. There is one course of toxicology.
Oh, you have that. Excellent. Because we're finding, you know, the more background, even, you know, that pharmacy students have on the drug development process and what goes into that helps a lot. And, you know, it speeds up training when they start working in industry and things like that. So very, very important.
Thank you.
So that's great. So thank you so much. I think that I don't see more questions for the time being. So
oh, good.
I think that yeah, because we were a very tough question. Very, I mean, serious question. So very, very tough question. So thank you so much. I just want to say thank you again. You have the contacts. We will, as usual, we will put this the video in our channel and in the PTS website and Granada Salud website. I hope that we will keep working on this because it's really paramount and we need a lot of advice and expertise here. So thank you again. I will say
Lourdes.
Yeah.
Lourdes, I wanted to to point out a couple of things that Paul raised. Yes, three things. One, he said that well, he loves working at Covance because every day is a challenge. So I cannot agree more on that. And I think that we all share the same the same feeling. So every day is a day that you are learning something new, and we love challenges. And this is related to what I explained at the beginning. So just two points that I wanted to clear up. So we do work with consultants. Paul already mentioned that. This is something that we do with great pleasure, and actually we work with a bunch of consultants on a regular basis, and they are bringing their customers all the time. And I also wanted to point out that on that topic that there are some companies, small biotech, that might be frightened or scared to work with a big company, with a big CRO as Covance. So we love to work with small biotechs. Actually, this is my job to find out and to reach that small biotech. And the trend lately is to start discussing even before they become a company. So it's never too early to discuss. So anyone who has a project, an interesting project that they think they have any doubt, any question, so we are more than happy to talk, to discuss. Don't be shy. Don't think that it's too early or you will make us waste our time. It's our job and we look to those who, okay?
So we really appreciate that because we think that it's very important and we need to begin developing the regulatory strategy when you move from discovery to development or in license. So it's really a pleasure and a great opportunity to have you on board. And we know I want to say something that also that Covance is helping us with our accelerator that we are working here at PTS and are providing some kind of service too. So we really appreciate your time and consideration.
Thanks to you, Lourdes.
Pues thank you to everyone. See you. Bye now.
It's a shame we can't go and visit you.
hopefully, because Granada is amazing, you know. I mean, it's a wonderful. I mean, we like our ecosystem a lot, you know, with the snow mountain here and all of that, and you know, with many good people, with a lot of science and with good ideas. And we have also people from all Andalusia. Joan Montaner is from Cadiz, and he's now a little farther, but hopefully, I mean, people will come here, and we will develop this ecosystem more and more. So thank you so much.
Okay, thank you.
Thank you. Bye-bye.
Bye-bye.
Take care.
Bye now.
Bye-bye.
Bye.